or injuries.
The neighbor girl fell on her bottom, cracked her bottom, ran screaming to her mom.
Nice.
Oh, we're on live.
Oh.
We're talking about Cameron's little bouncy toy thing.
That sounded kind of weird.
Yeah, their kids were playing this morning. They're inside. They're not coming outside. And one of the neighbor girls, she fell off and hurt herself. Anyways, I didn't even know we were live. The introduction to Paxlovid in December 2021 marked a pivotal moment in the COVID-19 pandemic. As an effective antiviral, it has successfully treated millions of patients. However, as with many antivirals, scientists recognize that Paxlovid may eventually lose some of its efficacy due to the emergence of drug resistance. We all have that.
Immune system, right?
That's right.
Immune system. Sure.
It's a good thing for our bodies.
It's one of the few drugs that I hear being advertised still on, like commercials or whatever.
For COVID, maybe?
Yeah.
Oh, yeah.
I mean, there may be others, but that's the only one that I seem to see on a regular basis.
So to address this challenge, researchers are exploring innovative approaches to combat SARS-CoV-2 and similar threats. Scientists have pioneered antivirals targeting viral methyltransferases, offering a new strategy for treating RNA and DNA viruses. This breakthrough could complement existing therapies, offering robust solutions against future pandemics. Would you like to know more?
So a recent study from the Tushel Laboratory has unveiled a groundbreaking proof of concept for a new class of antivirals. These compounds target a specific type of enzyme critical not only for SARS-CoV-2, but also for a wide range of RNA viruses such as Ebola and dengue
fever,
and systolic replicating DNA viruses, including pox.
The pox
viruses. We don't want them to come back.
Nightmare Before Christmas. The pox.
Yeah, we don't want them to come back. This discovery could lay the foundation for more rapid and effective responses to future pandemics, potentially offering broad spectrum solutions against diverse viral threats. Quote, "Nobody has found a way to inhibit this enzyme before," says Thomas Tushel, the FMA, is that AL? A1.
Al-Aql?
Al-Aql. Oh, is this his name?
I guess.
Al-Aql and Margaret Al-Aql, okay, professor at Rockefeller. Quote, "Our work establishes cap methyltransferase enzymes as therapeutic targets and opens the door to many more antiviral developments against pathogens that until now we've had only limited tools to fight."
So if the Black Plague, the Black Death comes back,
we don't want that.
We might have something to fight it.
Yeah, well, let's hope we don't have to. That's an
true.
ugly one, right?
Very ugly. Billions died in that one.
Wiped out millions.
Yeah. The way so many RNA viruses thrive is by modifying their RNA caps, specialized structures that stabilize viral RNA, enhance its translation, and mimic host mRNA to evade immune defenses. That reminds me of Moderna. Oh, I'm thinking of Moderna, mRNA. Okay. RNA capping relies on enzymes called methyltransferases. I'm trying to say that, man. Making it a tempting target for antiviral therapies. But most antivirals, including Paxlovid, instead focus on disrupting proteases, a different class of viral enzymes that break down proteins. Proteases, I guess.
Yeah.
Largely because these enzymes were previously targeted and prevented viral spread. Quote, "Inhibiting methyltransferase required using a non-conventional RNA substrate, adding a new challenge to drug to discovery," Tushel says.
For Tushel, an RNA expert whose work has already led to multiple RNA therapeutics for treatment of genetic disorders, that was not much of a complication. After he restructured his lab during the pandemic to focus on antiviral drug discovery, Tushel realized that there were clear advantages to looking beyond protease inhibitors.
Yes, he saw an opportunity, restructured his lab.
Yep, good deal. Tushel suspected that viruses would be less likely to dodge a combination therapy that targeted two unrelated viral enzymes at once, such as protease inhibitor alongside a methyltransferase inhibitor.
Hmm.
He also realized that drugs targeting viral methyltransferase, distinct in structure from the human enzyme, will be highly selective and not impair human enzyme function.
That sounds promising.
Yeah.
In search of a molecule capable of inhibiting the SARS-CoV-2 methyltransferase (NSP14), his team screened 430,000 compounds early. That's a lot.
That is a
In
lot.
the pandemic in the university's Fisher Drug Discovery Resource Center, and discovered a small number of compounds that inhibited the viral cap methyltransferase (NSP14), a multifunctional enzyme with methyltransferase activity.
Okay.
Hence the name.
Yep. Those compounds then went through an extensive chemical developmental process to create optimized drug candidates in the partnership with the Sanders Tri-Institutional Therapeutics Discovery
A lot of
Institute.
hands in the pots here.
Yeah, these are some long names.
Hmm.
Maybe they get paid by the letter.
Maybe.
Compounds with improved biochemical inhibition were then subjected to cell-based assays conducted by researchers led by Charles M. Rice, who heads the Laboratory of Virology and Infectious Diseases at Rockefeller. Finally, colleagues at the Center for Discovery and Innovation in New Jersey then tested the compound in our favorite lab animal,
Mice.
under BL3 safety conditions, whatever that is, and demonstrated that it could treat COVID-19 on par with Paxlovid. Wonderful. Tushel and colleagues also demonstrated that the treatment remained effective even if the virus mutated in response to it. That's another good thing, right?
Yeah.
And that there was no synergy when combined with protease inhibitors. Quote, even in isolation, a virus would have trouble escaping this compound, Tushel says. Again, another quote. But as combined therapy along with a protease inhibitor, escape would be almost impossible. I like that. If I'm understanding this correctly,
Yeah.
so you're not going. This is not going to build it. You're not going to build up an immunity to this thing, right? It's going
Right.
to keep on fighting.
Yeah.
So
The findings not only validate viral methyltransferase as promising therapeutic target, but also suggest that Tushel's particular inhibitor would have minimal side effects. Quote, the
like
mechanism.
that too.
Like what?
We like that too. Minimal side effects,
I
right?
thought you said tattoo.
No. We like that too. Also, we like that also.
The mechanism by which the drug acts is unique, he notes. In fact, the compound takes advantage of the unique structural features of the viral methyltransferase. They keep saying that word. I do not think it means what they think it means.
What are we talking about?
Methyltransferase.
Okay.
Also requiring the presence of the reaction product of the methyl donor SAM, meaning that the lab's compound selectively targets the virus without disrupting human processes. Well, that's good.
Sounds like a good thing.
Yes.
Yep. Quote, we're not ready to test the compound in humans, Tushel cautions. Probably the mice are probably pretty bummed about that.
Yeah.
An ideal clinical candidate needs improved stability, bioavailability, and a series of other pharmacologic properties that remain to be optimized in the long term.
Ooh.
We're in an academic lab. For that, we need an industry partner.
So it sounds like he's asking for an industry partner.
Yeah. So who are we talking about here? Like big pharma?
Big pharma.
Oh, yeah. No, we don't want that. Private money. We need some private money in there. Just Elon, get in there.
There you go.
You know,
Vivek.
Throw a couple billion to these guys. And
Cuban.
See what you can help them.
Mark Cuban.
Yep.
No.
Yeah. Maybe
Bezos?
Maybe he can be one of the lab rats.
Okay.
Or mice.
Okay.
In the immediate future, the Tushel lab is expanding this work to explore inhibitors for RSV, flaviviruses such as dengue, and Zika.
Zika is like that. If you get a mosquito that bites you and you're pregnant, the baby is born with this little head, a big body. It's bad if you're pregnant. You get
Okay, we don't want Zika then.
Yeah.
As well as mpox and even fungal infections, which all share a similar enzymatic vulnerability. Quote, this work opens the door to targeting many pathogens, he says. It's a new opportunity to prepare for future pandemics.
Yeah, if they can wipe out Zika, I'd be happy with that. That's
I've never heard of that.
oh yeah, the baby gets deformed.
So
His head is so
So
small.
where does this happen? Third world
South America, yeah,
South America.
Brazil, like
So is
type
this
area.
why is this transferred through mosquitoes?
Mosquitoes, yeah.
So this is why the
Zika.
big push for the mosquito netting and stuff,
Yeah,
right?
yeah,
Okay.
yeah.
All right.
So just want to share. We got a little bit of time. Share a sad story I saw on YouTube talking about mice. It's near Cedar City. This lady came upon
Cedar
this.
City, Utah.
Yeah,
Oh.
a golden eagle on the side of the dirt road. Back in the way back, and it was like hunched over, and so she got this conservationist who has 50 years working with hawks and birds of prey and stuff. Came out. They took the little guy. He took him, put him in a cage, took him back to his house, got a little mixture together, and got it down his throat and got him to drink it. And then he took a plate full of mice with the mixture on it. And, and you can see in his eyes, he was gone. He was breathing, but he was gone. And he was, taking the mice, and they fed him like about 10 mice, right? And then, okay.
All at one time?
Yeah, like, put it in, and then they gulp it down, and then put it in, gulp it down, about 10 of them.
Wow.
Right? And then, so you'd think, okay, he's got something in his belly, he's got liquid, so they left him for an hour, came back, put some more liquid in him, and then they showed the segment in the middle of the night, same thing, they liquid and more mice, like about 6 or 7, and he didn't look like he was, really wanted he looked like he was gagging to get rid of it, and he had to push it down. I'm yelling at the guy on YouTube, I'm yelling at him saying, let him go, just let him swallow it, give him a chance, and he's like pushing it down. That golden eagle died over the night. It was so sad, but with mice, I mean, it's like he gave up. It was so sad. He was on the side of the road.
Gave
big
up.
Old
well,
talons.
Did they say what happened to him?
No, the lady found him. Just slumped over. Called conservation. They came out to try and save him, save his life. And the guy said he's been doing this for 50 years. And what he said, normally people who do this after about 4 years, they stop because it's so sad. All the birds of prey that they lose. But he's got a buddy, and he had the buddy with him, the eagle buddy, and he was petting him and loving him and saying, "This guy helps me get through the darkest times," and it was pretty cool, but it was so sad.
Stuff happens,
Talking about mice,
it's too bad.
It's a circle of life,
Right.
Beautiful, beautiful bird. All right, folks.
Time.
Yep. Thanks a lot. We'll see you on the next one.
Take care, everyone.
Oop, I missed it. There we go.